Hello and welcome to the Science for Societal Progress podcast. For this episode, I talked with Nuno Henrique Franco about animal experimentation. And we address questions like, why do we do animal experiments at all? What can be done to reduce the amount of animal experiments? What are the regulations scientists have to comply with in order to be even allowed to do experiments on animals? And what are scientists doing to communicate while animal experiments are being done and why transparency is important? And if you want more information, don't forget to check out the show notes on our website, www.scienceforprogress.eu. It includes a summary of this episode and resources for further reading. I am your host, Dennis Eckmeyer, and you're listening to episode 15 of the Science for Societal Progress podcast.
My name is Nuno Franco.
Nuno is an assistant researcher at the Institute for Investigation and Innovation in Health, for short I3S, in Porto, Portugal. And he is an expert in animal well-being and scientific research.
So I've worked on animal welfare, animal ethics, regulation. I'm also currently coordinating the national network of animal welfare bodies.
Animal research is done mostly in context of health research, but why do we use animals?
Animals are used as proxies. We couldn't do it in humans. We couldn't do it from an ethical perspective, but also from practical perspective. It would be impossible to recruit the sheer number. We couldn't standardize the animal line, standardize the conditions if we were to do it with humans. And of course, some experiments are indeed somewhat invasive, and this would raise significant ethical issues.
So we use animals as models for human physiology, because human experimentation at this level would be unethical. And even from a practical viewpoint, humans simply aren't good study subjects for physiological studies. How well do findings from animals translate to humans? Are animals a good proxy for humans?
History has been proving that in most cases they are, but there are several instances where we could do better, we could improve our models. It's an ongoing debate that has several layers that involve social issues, ethical issues, scientific issues as well.
We all agree that the use of animals should be reduced and that there needs to be an effort to do that. The principle applied internationally is commonly known as the 3 R's: replace, reduce, refine. Replace means to replace animal experiments where animal-free methods exist. Reduce means to always look for ways to obtain the same information with fewer animals or more information from the same animal. And refine means to optimize your methods to cause less harm. So less pain, less suffering, and less distress. When it comes to replacement, there are claims that we could get rid of all animal research. Is that so?
Here we have to do a distinction between animal testing and basic and applied animal research. When we say animal testing, we talk about the testing of toxicity, of efficacy of a given drug or any given compound. And this is much easier to standardize. We have increasingly new alternative models. I don't like the term alternative because it means that it's the second choice, when indeed in some cases these models are actually better, either because they're cheaper or they're faster or more ethical or all of the above. So, but non-human methods, whereas for basic research in which we are still trying to understand the underlying mechanisms of what happens in the animal, we can't model what we do not know. We cannot model a full living animal being and all the interactions between all the systems. If we could, then we had reached a point in which we wouldn't need to, because we would have known everything about the biology of that animal.
In other words, we have become better at replacing tests, for example, tests on how toxic a compound is. by using animal-free methods like cell culture. But when it comes to physiological research, complete replacement of animal experimentation would mean that we have learned everything that there is to know. We are still a very long way from this to happen. However, there are of course ways to conduct some experiments without having to use whole living animals. One relatively new development are
three-dimensional organoids. So very complex, small models of that are not just lumps of cells, but that already has some structural and physiological complexity that mimics a full organ. And we can also link by microfluidic systems, link several organs in order to simulate a living being. And I think that technology is quite promising, even for basic and applied research. But we are still far from being able to stop using animals.
In fact, the outcomes from organoid experiments will still have to be verified in the whole animal. But overall, the use of organoids could reduce the use of animals. But organoids are not for everybody. I'm a neuroscientist, and for neuroscience, these organoids are hyped as quote-unquote mini-brains. To my knowledge so far, these organoids don't even come close to the intricate complexity of a real brain's microstructure. Furthermore, if you recreate a functioning brain, even a small one, new ethical problems would arise about the ability of the organoid to suffer. But this is science fiction. At this point, organoids are no replacement for most of neuroscience at all. Another important point is the cost. How do animal-free testing methods, we are not talking about fundamental research, compare to animal experimentation?
Well, in terms of developing costs, developing and validating a model costs a lot and takes a lot of time, because we have to be sure that it's a proper model and a better model than we had previously. But once you have it, it's much easier to produce and to keep and to stock and to use, and you can do high throughput testing. So let's say that you want to test 1,000 compounds, rather than making 1,000 animal tests, each test with dozens of animals that occupy a lot of space and eat, and you have to have special facilities and special trained staff. You do it in very small, like biochips, let's call them, and you do a ton of them. So once you develop and validate the model, becomes much more affordable to use these non-animal methods in many cases, at least.
Oh, this is great news. As you maybe know, I've been an animal researcher up until very lately, and I actually also think the fewer animals we have to sacrifice, as it said, to science, the better. Okay, so we established that replacement of animal research with animal-free methods is possible, but only to a certain degree. There are still scientific questions that can't be answered without animal experimentation. If a researcher finds that their research cannot be done animal-free, they still can't simply find themselves some mice and just start experimenting. There are still 2 of the 3 R's left: reduce and refine.
You have 3 licenses to do an animal experiment. So you have the license of the user, so of the researcher, and for you to obtain that license, you have to have extensive training. You have to have the license for the project. So there's a harm benefit analysis of your project. So you write a detailed description of what you intend to do with the animals, what is the severity that you expect your procedures to have, and then some harm benefit analysis will be made by the competent authority. So you need that authorization as well. and you have to have an authorization, a license for the institute, for the establishment, they call it in the legislation. And this means that you have to have really well-equipped, safe facilities. All your staff has to be trained as well. You have to have a veterinarian on call every day following the animals. You have to have trained technicians. So there's this ecosystem of competent people that are ensuring that you comply with legislation, and also there are regulatory mechanisms to ensure that you have all of these in place. So the 3 licenses, and of course that you comply with everything in order for these licenses to be in order.
Mice are the most used vertebrate animal in research. What does it take to do experiments on mice?
The principles that you apply when you do research with mice are the same that you'll apply with any other vertebrate, and including cephalopods, which are actually invertebrates, but are also included in the legislation. Only primates and companion animals have, let's say, a different statute in the legislation, but 90% of the regulation is the same, or even more. So, in order for you to get a license to do your project, first you have to justify why you are doing the experiment, why that mouse is needed, why you couldn't do with a non-animal method. And in many cases, what happens when I actually look at a lot of projects, what happens is that you do have non-animal methods in place, but at some point you will move on to using the animal, but you have to justify that that the use is well justified. So I apologize for the redundancy. And also you have to classify the severity, as I mentioned previously. You have to classify the severity of procedures. So we classify the severity of procedures in mild, moderate, or severe. There's a somewhat separate category therefore in which animals are completely anesthetized, and everything that happens to them is under terminal anesthesia, and the animals do not recover from it. And so I tend to consider that category separate. But you have mild, moderate, and severe. So mild is when you inflict mild harm, and this can be drawing blood, or an intraperitoneal injection, or just containing and doing some behavioral research. You have, but if it's mild, it has a mild impact, but for a long period, then it becomes moderate. The same thing for moderate. If you have a moderate, if you inflict moderate harm or suffering, but if it is short term, then you classify it as moderate. But if it's long term, then it becomes severe. And then you'll ask, what happens if your procedure is not just severe in the short term, but severe in the long term? Then you are not allowed to carry out that experiment. Any procedure that results in long-lasting, unameliorated harm is simply not allowed. You can apply for an exception to the European Commission, but so far I haven't heard of anyone who has done it. You also have to write a non-technical summary, and these are available online. So the Portuguese people and every citizen in the EU can consult in their own language the non-technical summary, which will describe the severity of the procedures, why the animal is being used. And then you have to ensure that every single person who will be in contact with an animal has been thoroughly trained and has a certificate and a license by the competent authority. So all of this has to be in place. And in some cases, the competent authority will ask, will tell you that they will carry out a retrospective assessment. Meaning, so if you prospectively, because it's always prospectively when you've not done the project yet, when you prospectively classify your project as severe, or if you use non-animal, non-human primates for any severity, then the competent authority will do a retrospective analysis and try to verify whether the severity was exceeded, whether the benefits that you proposed were achieved, and do a reevaluation of the harm benefit. So there are a lot of steps prior, during, and after, in some cases, the project. So there's a lot of scrutiny.
Right. So these levels of severity, how are these defined? Do they look at the animals and the state of the animal, or is it just, oh, it's just an injection, so we don't care? It's a light thing.
Yeah, this is quite a big issue because there's a lot of subjectivity. What is long term? I mentioned long term harm or suffering, or short term harm or suffering. What is long term? What is short term? So you have to base your evaluation with you have to base it on experience, on the experience of the vet, on the experience of the animal welfare body that know the outcome in terms of the clinical signs that the animal will manifest. You have to study the phenotype of the model that you use. Let's say that you have a model with a deleterious gene mutation, and you have to know disease progression and understand at which point the animal will start manifesting signs. There's a number of workshops, and I've been training researchers on these workshops on how to classify the severity of procedures. We're trying to harmonize severity classification across Europe, but it's really hard because of the subjectivity and cultural differences. And even species, the empathy that you have towards a dog or towards a mouse or towards a fish, which is also a vertebrate and will also be protected under the legislation, will be different. And how you assess the severity for that specific species will tend to vary, even if you do the same thing. So you basically you base a lot of your assessment on previous experience. There are also a number of published examples. So you have Annex 8 in the directive, you have guidance documents in the European Commission's website, and this group by Falasa, Eslav, and Eklam, in which I was also working on. We developed a few other examples. So we've published this year 12 more examples with different species, with procedures of varying severity. We have to have a lot of factors into consideration. Let's say if you work in the wild, you have to consider capture, for instance. If is tagging the animal considered husbandry, or is it contemplated when you are measuring the cumulative effect of a procedure? So all of these details have to be sorted out. So this is why we are having training and trying to harmonize across Europe. And Falasa has been having a lot of work in this regard.
Since we are talking about ethics, Nuno shared with us some preliminary work he was doing on the ethical values of researchers in respect to animal experimentation.
Now, I am currently in bioethics, in animal ethics. What I'm currently researching, this is somewhat of a side project, but we've been actually trying to understand the attitudes of researchers towards animal tests, animal research, and trying to identify which factors weigh in their consideration of replacement alternatives, of severity, of what species they would choose, and we were also offering them dilemmas to sort out. And what I've realized so far is that us researchers are not that different from the general public, in the sense, for instance, that species matter. It shouldn't, well, objectively, it shouldn't matter. So the suffering of a dog shouldn't be different from the suffering of a pig or the suffering of a mouse. But when you ask researchers and the severity is high, let's say, or the benefit is not perceived as high as in another setting, then researchers will do an ethical evaluation on a case-by-case basis, the same way that students will. So there are studies with students, with biology students, there are studies with the public in which they propose different case studies, varying the severity and varying the species, and the results completely reflect what you see by the general public. So from an ethical point of view, what I've picked up is that we are utilitarian, in the sense that us researchers, tend to be utilitarian most of the times. So in the sense that we do a harm benefit, a cost benefit analysis, in the same way that the directive proposes institutes and competent authorities to do. But we also take into consideration the relational aspect. So the extent to which we can establish a relation with the animals. So companion animals and primates are also given special consideration by researchers. So we're not fully utilitarian. Also, some of us will also have some, not extreme, but some moderate animal rights perspective, in the sense that you'll see some researchers say that doing this or that violates the right of the animal. And I wasn't expecting that from researchers. I was expecting researchers, scientists, to be more objective, less subjective in their analysis, and it turns out that they're the same as everyone else.
Well, that's good to hear, right?
Yeah,
or should they be better?
You won't find any extremists, I think. But yeah, it's quite diverse. Mm-hmm. Actually, I was quite surprised to find that, and even some, for instance, scientists, the same as the general public, will think it has more merit to study, let's say, a cancer than obesity. Or drug addiction, even though there are several factors other than people's choices that drive to obesity and drug addiction. So they'll make that moral judgment. So we are not immune to that, and that's very interesting. And this is one of the reasons why I'm also interested in ethics and regulation, and trying to get to the core of our attitudes and our values and understand what makes us human. And in a sense, I think caring for other species is, in a way, something that makes us human. It's a human trait, isn't it? If you think of it.
Yeah, I think so. Yeah. So yeah, this is really interesting research into the ethics of researchers. And was that focused on biomedical researchers or just scientists in general?
Actually, our sample was made of people who were either working with animals or who intended to work with animals. So it was biased in that sense. And this is one of the reasons why I expected attitudes and values to be somewhat different. And they were only in the sense that accepted that animal research was necessary. But other than that, all other factors that intervene in the discussion and the ethical and social debate surrounding animal research are there as well.
Oh, that's good news, I think. Nuno found no difference in the ethical reasoning about animal research between the general public and people working in life sciences, except maybe that they all agreed that in the end animal research is necessary. I was wondering if the fact that some members of the public reject animal experiment outright would be because they don't know enough about it.
It depends on whether you ask them, you stop someone on the street and you ask them, what do you think? Are you pro or against the use of animals for testing, I don't know, chemicals? And a lot of people will tell you, I'm completely against it. But this is what we call a cold start question. But if you start your question with animal research has so far been quite helpful in developing vaccines and therapies for a number of diseases that affect humans and have been quite useful and important for the development of science and medicine. And then you ask, what do you think of the use of animals? Then with this warm start, then people will be much more moderate in their views. However, there is this small segment of society that will completely oppose any use of animals for any reason. And I think it's a valid point of view. And I would say this segment of people is around at most 5%. But they will not do an utilitarian calculation between harms and benefits, and they will have a very hardcore animal rights stance, let's say. Because if you grant animals the same rights as humans, then you can't do anything to them without their consent. Whereas a human can consent, an animal can't consent. And any animal use for food, for work, for research is out of bounds for these people. So in terms of outreach, you know that it will be quite hard to reach to those people. But I think most people, going back to your question, most people are moderates, unless you do this very cold start question. Do you agree with animal research? Then at least more than half of the people will be against it. You have to provide some context, and this is what we with the outreach are trying to do, is provide context.
Providing the context of the usefulness of animal research is one thing outreach can do. Another way is to ensure that the public can see for themselves what is happening. One mechanism we already mentioned is that researchers have to provide summaries of their research proposals in non-technical terms. These are available online. But more is being done in terms of transparency. So you've been working on a so-called transparency agreement on animal research?
Yes, I was somewhat involved as a member of the board of the Portuguese Society for Laboratory Animal Science. That together with the ERA, the European Animal Research Association, we had this transparency agreement between the 16 most prominent research institutes and universities in Portugal, in which we pledged to be more transparent on why and how we use animals in science. I think transparency is a much better approach than trust us.
Yes, yeah.
Blindly.
No, of course.
We are scientists. We all have PhDs. You should trust us.
Yeah, that's not good.
I think trust has to be earned. And in some cases, we are not angels. There are a few instances that things go wrong, and these people have to be called out, and sanctions have to be in order, and sometimes completely forbid some people of working with animals, and some people shouldn't be working with animals. But overall, I would say almost all researchers working with animals are compassionate, competent people. And the public has to know that.
So let's sum it up. Animal research has been tremendously useful for the improvement of our understanding of life and led to advancement in medical and also veterinarian care. Animal testing of compounds can be more readily replaced with animal-free methods than scientific research experiments. In fact, once new testing methods are validated, they are often even cheaper and easier to implement. For cosmetics, the European Union even banned all animal testing and also products which were tested on animals outside the European Union. But for animal research experiments, there is no way to replace them all with animal-free methods. We can, however, approach a question by finding out as much as possible using animal-free methods first, before we move to animal experimentation. Researchers have the same ethical reasoning and morals as the general public, and to ensure animal research is conducted humanely, they have to justify their methods and comply with strict regulations. These regulations implement the 3R principle. Replace animal research with animal-free methods if possible. Reduce the number of animals needed to gather the information that you need, and refine methods to avoid pain, suffering, and distress as much as possible. And transparency is increasingly important. The public needs to know what is happening in the laboratories. This is why researchers provide summaries of their funded proposals, and institutes conducting animal research are working on ways to give the public insight into their conduct. For a written summary and further readings, find the show notes to this episode on www.scienceforprogress.eu. If you have questions, critique or suggestions, get in contact by email info@scienceforprogress.eu or on social media @sci4progress for Twitter and Facebook. Science for Progress is free and accessible. However, in order to keep going and continue to improve and grow, we need your help. You can find information on how to support us on our website www.scienceforprogress.eu if you follow the menu to Your Support. However, the best way to support us is through Patreon. We are halfway to financing our monthly expenses, and it would be an enormous relief to have that covered. Check out the perks on www.patreon.com/sci4progress. My sincerest thanks to those who are already supporting us. Have a good day. Bye bye.